Hello everyone, welcome back to Zwitter Ions. Today we are going to discuss why amino acids are called as Zwitter Ions. So what is the reason amino acids are considered as Zwitter Ions? The first reason is it consists both positive and negative charge. So the consisting of positive and negative charge makes it a Zwitter ion. And next we can say it consists of both acidic and basic character. How you can define it is an acid? An acid can donate a proton, correct? And a base can accept a proton. So amino acid acts as an acid and base. It depends upon the pH. So pH regulate the acidic and the basic properties of an amino acid. So when you dissolve amino acid in water, the dipolar structure will get. That means both positive ion and the negative ion. And it is also called amphoteric nature. You can see in the structure the amino acid, it behaves as an acid. How? The NH3 plus group, it donates a proton, correct? So this amino acid acts as an acid, and in this case the COH minus, it accepts a proton. That means it is a proton acceptor and it acts like a base, correct? So next is, in this reaction the positive charge, it converted to uncharged, right? This one, the NH3 plus, it becomes NH2, so positive to uncharged. And in this case the negative charge, it become uncharged, that is COH, correct? So let's discuss about the PI. So what is PI, or isoelectric point? It is the pH at which the molecule carries no charge. That means, as I said, the amino acid will be in NH2 form and the carboxylic group in the COOH form. It will be no longer in the ion form, correct? Or the net negative or positive charges will be equal. So what will happen if the net negative and positive charges will be equal? So they will neutralize each other and they become again uncharged. So what is isoelectric point? The pH at which the molecule carries no charge, or if they have charge, the negative and positive charges should be equal, correct? And this is a very important concept. If the pH is below Pi, then it will form the net positive charge, and if the pH is above Pi, it will form the net negative charge. So now question is, how we can calculate Pi? So in practical classes you might have observed that in titration we have to our basis to see the pH range, correct? So let's say in case of glycine, it is an amino acid. So in the X acids, the OH minus, that is the base equivalent we are giving, and this is the range of pH. So when we are increasing constantly the pH, at a certain pH the net charge will be equal. There is no positive charge or negative charge, correct? So how to calculate Pi? It will be the average values of pK1 and pK2. Then again question is, what is pK1 and what is pK2? So pK1 is the pH at which the carboxylic group loses first its proton. Okay, so who will lose its proton first? Obviously an acid, correct? So carboxylic group is the first to lose the proton, and here the alpha carbon loses its proton. That's why the name is pK1. The carboxylic group is in alpha position, and the proton which loses, that is from alpha COH, correct? So this one is the pK1, correct? The COH group, it loses its proton, that is pK1. Then this point is the pi, where you can see the net positive and negative charges are equal. Is one positive, one negative, so it become on charge. So this point is the pi isoelectric point, and it will be pK1 will be 2.34 and pK2 will be 9.6. Then it will be approximately come 5.9. So then what is pK2? So pK2 is the pH at which the amino group loses its proton. So it is pK2 as it is the second group which loses the proton. The first group is the carboxylic group, that's why it is called pK1. And the amino group is the second group which loses its proton. So at this pH, it is called pK2. Okay. So this is all about the titration curve. When you are increasing the base, when you are increasing the pH at a particular pH, both the charges are neutralized and there will be no net positive and negative charge. That will be the pi value, and the pK1 is the carboxylic group, alpha carboxylic group, where it loses its proton. That pH is called the pK1, and the pK2 is the pH where amino group loses its proton. So this is all about the concept of Zwitter ion. I hope it will help you. Thank you very much.